Hello, everyone. Welcome to today's live broadcast of the first part of the three-part webinar series on digital RNA sequencing. I am Krishnan Alampalam, and I will be the moderator for the entire series. Before we begin, we have a few important announcements. First, all products shown here are for research use only. They should not be used for diagnosis, treatment, or prevention of any disease. For any information, please visit us at www.qiagen.com or talk to our technical support team. Number two is to ensure that everybody can hear the speaker clearly. We have muted all lines. So for any kind of interaction with us or the speaker, please use the questions box that appears on the lower right-hand side of the monitor. Just to make sure that you can hear me clearly, please enter yes or no in the questions box so I know that you can hear me clearly. Okay, thank you. Thank you. Now, today QIAGEN is very excited to announce the launch of QIAseq targeted RNA panels. These have been developed as a sample-to-insight solution for quantitative gene expression profiling using digital RNAseq. With the introduction of this new technology, there are many questions, starting with what is digital RNA sequencing? How does this differ from standard RNAseq? Why one should use digital RNA sequencing for gene expression profiling? How do you plan an experiment? To answer all these questions, we are very proud to announce this three-part educational webinar series on digital RNAseq. And this series is being presented by scientists from our R&D department who actually worked on developing this technology right from the beginning. So these are the leading experts in using digital RNAseq for gene expression profiling. To start with, let me introduce you to today's speaker, Dr. Eric Leder. Dr. Eric Leder is not a new person in this field. He is a pioneer in the field of RNA technology. He's the person who invented RNAlater, which is one of the basic reagents that is being used by all reagents in this field of RNA. He's a molecular geneticist by training and is currently the Senior Director of Product Development at QIAGEN. In his 13+ years at QIAGEN, Dr. Leder has led to the development of tools for functional genomics, gene expression profiling, microRNA profiling, and characterization such as the gene read targeted panels for next generation sequencing, and now the digital RNA-seq panels for gene expression profiling. With that, I will hand it over to Dr. Eric Leder to present his webinar titled Introduction to Digital RNA Sequencing.
Thank you for that kind introduction, Krishnan. Hi, everybody. I'm going to apologize in advance for this wonderful head cold that I brought back from the AGBT meeting on Saturday. So every once in a while, I'm going to punctuate my talk with a coughing fit or clearing my throat or some other strange noise, and you're just going to have to forgive me for that. Everything that we're doing here in this part of QIAGEN in Maryland is about targeted sequencing, and today, as Krishnan said, we're going to be talking about targeted RNA-seq, and what we're trying to do here is address the limitations of generally the limitations of smaller desktop sequencers that have limited budgets, as well as the challenges of sequencing from larger numbers of samples with a more targeted approach where you're looking at specific content rather than doing whole genome or whole transcriptome analysis. So if I can figure out how to advance the slides, life will be good here. Hang on. Yeah, okay. So What I'd like to talk today about is a little bit of an introduction to expression profiling and give you the 1-minute historical perspective. I've been doing gene expression analysis, I'm happy and somewhat sad to say, since 1981, so for quite a long time. Talk a little bit about whole transcriptome sequencing and then why we started to think about doing targeted expression analysis, and then do a much more deeper dive into the QIAseq targeted RNA-seq technology. Talk a bit about the incorporation of random molecular barcodes to improve the precision of targeted RNA-seq. Tell you about the advantages of our workflow that we've developed. Give you some proof data on the performance and the reliability of the process that we've developed. Take you on a bit of a tour of our primary and secondary analysis, and then give you a brief introduction about what you can expect from the second and third webinar, and follow up with some question and answer period. So maybe not the dark ages, but the dim ages. Back when I started doing gene expression profiling, I did my first northern hybridization experiment in 1982. And back in those days, you essentially had two choices when you were looking at gene expression analysis. Northern blotting, which was essentially named northern blotting as a joke, as the sort of the opposite of southern blotting. Southern blotting was named southern blotting for DNA hybridization, named after Ed Southern, and northern was a play on Ed Southern's name. It was a way to transfer RNA to a membrane, probe that membrane with a radioactive probe, and attempt to quantify the signal either through exposure to a piece of X-ray film. Later on, there were other methods to try to quantify the amount of signal on that membrane. It was characterized by being relative quantification, so how much signal was on that membrane. It was relatively low precision. The dynamic range was a few logs, a relatively small dynamic range. Assay throughput was rather low. We had one probe per membrane. Sample throughput was relatively low. We had maybe up to a dozen samples on a northern blot, and sample requirements were very high. So typically in a northern blot, you'd have your 12 samples, and you would be running from maybe 10 to 20 micrograms of total RNA, or you poly-A select it and run a microgram of poly-A RNA on a northern blot per sample to be able to detect a typical mRNA. Nuclease protection assays had somewhat better sensitivity and somewhat better quantification and precision than northern blotting. They required the production of radioactive probes, so they were a little bit more involved in the technical side of it. Gave you less information in terms of the size of an RNA transcript, but again, better precision, better dynamic range than a northern blot. You could do multiple assays per sample per lane, so better, higher assay throughput, somewhat higher sample throughput. Never really sort of had the popularity of northern hybridization because of the technical demands of having generally to clone a probe to prepare a riboprobe and that kind of thing. We then moved to RT-PCR when PCR was invented. Most people were doing relative quantification in endpoint PCR, running products on a gel, staining with an intercalating dye, trying to quantify the products, and it had a lot of allure because when you ran a gel and stained it with a dye, you got a nice sharp band on a gel. You can quantify that band. And it looked like it was science, but it was fraught with a lot of misinterpretation. The dynamic range was small. It was very easy to get the wrong answer with high precision and get the wrong answer time after time again. That improved later on, and we'll talk about that. And then there was a lot of use of filter-based hybridizations, also known as the dot blot, where you would do sort of a reverse hybridization and affix the probe, typically a plasmid or an oligo, to a filter and label the bulk RNA and hybridize the bulk RNA to the membrane rather than the other way around, and quantify the signal on the dot. It's essentially reverse northern. The attractiveness of it was that you always got your signal in a tight little dot. And if your RNA was terribly degraded and really ugly and your hybridization was terribly nonspecific, it still made a nice little dot, and you could tell your advisor that your experiment worked, when in fact mostly what you were seeing could possibly be noise and not actually the signal you were looking for. So qRTPCR improved things quite a bit. It has much higher dynamic range, so a qRTPCR experiment typically has 10 to the 6th or 10 to the 7th fold dynamic range, and very high precision. So you do technical replicates and you have reasonably good hands, you can get extremely high precision, not necessarily high accuracy, so you can get the same wrong answer all the time, again. You have moderate assay throughput, so you can, for example, in a PCR instrument, look at 384 assays in parallel from one reverse transcription from one cDNA synthesis reaction. These are typically still singleplex assays, so if you're doing 384 assays, you have relatively high sample requirements in that you have to split that sample into 384 individual reactions to do 384 different assays. Hybridization arrays, which are now typically oligos that are affixed to glass slides, give you, again, relative quantification like PCR with fairly good precision. The dynamic range is a little more compressed than PCR, but you have extreme high assay throughput. So you can do, for example, the whole transcriptome in one experiment, but the experiments are relatively low in sample throughput. So you're not doing typically thousands of samples. You're doing a relatively small number of samples at a time, but looking at lots and lots of targets. And people are still obviously still doing hybridization arrays as screening tools, finding candidate genes that are interesting for their system, and then looking at the differential expression of those genes in higher sample throughput, still typically by qRTPCR. Another fairly recent technology that people are using for extremely highly accurate quantification is digital PCR or droplet PCR, which has a very broad dynamic range and is very sensitive, where you can very accurately quantify even extremely small numbers of target. It has relatively moderate throughput for assays and relatively low sample throughput. It's not really applicable to looking at lots and lots of different assays. So it's all of this leads us to whole transcriptome NGS, which is where today's discussion really starts, where we're looking at all of the transcripts in a cell by next-gen sequencing. And what we're doing is essentially relative quantification with very high precision. So we're sequencing and characterizing everything that's in a cell. If we sequence deep enough, we get very high dynamic range. If you want to call it assay throughput, we have extremely high assay throughput. So every target that's in that cell that can be identified and sequenced is identified and sequenced. Because of the cost, we're talking pretty low sample throughput unless you're a large sequencing center. You're typically doing relatively small numbers of samples. And because you need to dedicate a large sequencing budget per sample, because there's a whole transcriptome to deal with, the price per sample is relatively high. So this limits the number of experiments that can be done and the number of labs that can essentially afford to do these kinds of experiments. And typically, for me, they would be the first pass of an experiment, characterize everything you can in a relatively small number of samples, identify things that are of interest to you, and then find a technology that you can use to hone in on the targets of interest in a larger number of samples. So the benefits of whole transcriptome sequencing are that you can quantify and characterize all the RNA in a cell. You can identify all alternate splicing events. You can detect expressed SNPs. You can detect somatic mutations if they're expressed. You can detect allele-specific expression patterns if one or the other allele is expressed preferentially in a cell type. There's large computational requirements. You generate a massive amount of data. And as you see on this flow chart, we're typically taking mRNA or total RNA. We're fragmenting it, we're putting linkers and amplifying it, and we're sequencing all these small fragments and using our bioinformatics neighbors down the hall to put all this stuff together and to figure out what was in there in the first place and try to figure out what the relative amounts of all these different transcripts were. You're running these on high throughput instruments, which limits this to a core lab. It helps you develop relationships with skilled bioinformatics scientists. And it is not a simple task to do quantification on transcripts. It's very difficult to know that you're getting accurate estimates of levels of transcripts from different genes. So if you're only interested in gene expression, and quite a number of people who are looking at gene expression are actually only interested in the relative levels of gene expression between cell types. Are the levels of gene expression changing? A lot of the benefits of whole transcriptome sequencing aren't really benefits for you. So if you're not interested in characterizing RNA, splice variants, or post-transcriptional modification of bases, or SNPs, or allele-specific expression patterns, or you're not interested in all RNAs, and you're interested in a subset of RNAs, and you're only interested in expression of those, then a lot of the benefits of whole transcriptome sequencing are not really benefits for you. And you're spending a lot of time and effort hanging around with bioinformatics scientists when you could be screening more samples for less money and less time. And so that takes us to targeted expression analysis. And so there are potential advantages of applying targeted gene expression profiling. And you can see the word potential is in italics because these are all theoretical and they're real if you execute properly. So what we like to do is use the read budget of our sequencing machine, and that read budget is based on the instrument we're using and the chip we're using. So for example, an Illumina MiSeq has several different capacity chips you can run on it, and an ION Torrent personal genome machine has several different chips with different capacities you can run on it, and that capacity is fixed for that chip. So you can spend that read budget to read, for example, 100 genes or 1,000 genes, or 1 sample or 100 samples. So there's some thought that goes into how you design that experiment, and you'll hear more about that in our next webinar. But the idea is that you design your experiment to make the best use of that read budget so that your cost per data point, the cost per the things that matter for you, is maximized. It's also a quicker experiment because the runtime is quicker, and I'll explain why that's true. And the analysis is much quicker because we're only sequencing and quantifying and analyzing and putting together the data for the targets that we're interested in. And the sample throughput is much quicker because we can multiplex together on a desktop instrument easily 100 samples and up to several hundred samples, depending on how big the gene panel is that we're using. And as I mentioned, desktop platforms can be used for RNA analysis. We routinely use the PGM and the MiSeq for reasonable experiments, and the NextSeq for actually fairly large experiments. So you don't, for example, need that core lab across campus. A research lab-size instrument can be used for fairly large experiments. The NextSeq has an enormous read budget for a fairly reasonable price. The bioinformatics can be much simpler. So when I talk about targeted expression analysis, what I'm talking about is counting. And as I'll show you on upcoming slides, what we're doing is reading enough of an mRNA to definitively identify it as coming from a specific gene and uniquely tagging it so we know it comes from a unique transcript. So we don't need that bioinformatics guy down the hall to figure out what this sequence is. In addition, we do essentially no sample pre-processing, so there's no ribosomal depletion or blocking or poly-A selection or anything like that that goes on. You're using total RNA. And for a typical panel, we're using nanogram quantities. Our default amount of total RNA that we use for a panel is 10 to 20 nanograms of total RNA. You can use less, you can use more, and as I'll show you, when you're using random molecular barcodes, you know exactly what you've got. So you can use picogram amounts of RNA. You will make enough material to make a library and sequence it, but what you find when you're using unique molecular barcodes is that the number of molecules that you're characterizing when you use 100 times less RNA to start with is 100 times less molecules. So you have to ask yourself whether you're happy characterizing 100 copies of a gene transcript or one copy of a gene transcript. The sequencing doesn't lie, essentially. And finally, I'd like to consider the question of when would you use targeted RNA-seq and who would use it and why would you use it. So I think that there's two basic reasons you would be using targeted gene expression analysis. And one is if you've got a known gene list or a known pathway. I'm interested in genes that are regulated in cancer. I'm interested in genes that are regulated in development. I'm interested in genes that are regulated in this signal transduction pathway and this differentiation pathway. So if you know a particular pathway that or process that you're interested in, you either have a gene list in mind or we've already developed that list for you. Or if you've done a broader experiment, such as a large microarray expression analysis experiment or a whole transcriptome experiment, and you've developed a list of genes that you'd like to look at further, you can, for example, rather than do one sample at a time, you can now look at 100 genes in 384 samples at a time, and do the analysis right off your instrument onto our website and look at 400 samples a day. So this brings us to QIAseq RNA, which we call high-throughput digital NGS. From the beginning, we've designed this to be extremely simple to use, which means the complexity is hidden behind the scenes. We want users who are not necessarily experienced NGS users to not be intimidated to use this product, and we try to approach the concept of essentially peel and use, or open the box and use, as closely as we can. The system is a complete integrated system. You start with an RNA sample and you end with your data and even with a molecular insight. We're providing links to Ingenuity IPA analysis for you to do your first experiments in pathway analysis with this product. And some of the features of this integrated solution is that it's sensitive and highly specific. We're extremely flexible in experimental design, and we can help you guide you in thinking about your experiments and what machine you have in terms of how many samples and how many assays you're multiplexing together on your instrument. It cannot be simpler for you to address your system bioinformatically, and I'll show you that in an upcoming slide. And I mentioned before, there's no pre-treatment of your RNA, and we're making best use of your limited NGS budget. And in fact, the molecular barcodes that we're adding really lets you tailor your experiment to use your instrument's innate budget as best as possible. And it lets you leverage our know-how in terms of content. We have been developing gene panels for years and years, and all of that can be translated to NGS. So we have disease and pathway-focused panels, and our library panels are easy to modify for you, and you can also do fully customized panel content. Our panels are platform agnostic for both the ION and Illumina platforms, so PGM and Proton, the MySeq, the NextSeq, and the HiSeq. We incorporate the random molecular barcode into all of our molecules, so when you're counting RNA, when you're counting reads for a particular gene, you know exactly how many original RNA molecules you're sampling. So you know, for example, when you've got 30 reads, that they are 3 reads each of 10 original RNAs. It's a streamlined one-day protocol. It's easily automatable, and we incorporate multiple reference genes, which we've developed from our expression studies. and we incorporate genomic DNA controls to monitor in case there's something terribly wrong with your RNA sample. So we're trying to engineer robustness into the system so that your results are both precise and accurate. So some of the considerations that go into this, we do recommend for experiments in which you're biologically manipulating samples that you should do replicates. There's a lot of variability in biology, variability in life. So for robustness, you know, as in anything else, we recommend biological replicates. Technical replicates are generally not required. You are sampling thousands, millions of molecules. Once you get the hang of this, you see very little noise between technical replicates. We don't cover across a transcript. We're designing amplicons to a common region of a transcript for each transcript. We're doing multiplex PCR, and we're giving the bioinformatics people leeway in picking a region of the gene that is very constrained for their algorithm for primer design. So all of our amplicons are very constrained in size and TM and in PCR efficiency, so that we do extremely uniform multiplex PCR. So that works really well for us. But we don't generally, and we generally cover all variants that are in the ensemble database. In terms of sequencing depth, we don't express it quite the same way when we're using molecular barcodes as people generally do. We need to capture enough unique barcodes, and I'll show you this in an upcoming slide of each transcript, such that statistics make sense for us. And what this turns out to be is that as long as we can capture 10 unique barcodes per transcript per gene, the variance is very low. So I'll say that again: as long as we can capture 10 unique transcripts per gene, we're golden. It doesn't actually matter how many times we sequence those unique 10 transcripts. So whether we read them once or whether we read them 20 times, we're good. But we need to read 10 per gene. So if it's a rare gene, we need to read the whole population, maybe deeper. We don't do a stranded library prep. The amplicons are designed to not overlap with known long non-coding RNAs. So we're not reading, by design, not overlapping with semi-overlapping long non-coding RNAs. And the amplicons are short, so we don't actually have to do paired end reads on an Illumina or on a PGM. We're doing single-ended reads. We're getting through sample barcode, random molecular barcode, and the sequence itself with a single-sided read. The system is compatible with any upfront RNA that's isolated with a QIAGEN kit. So freely circulating nucleic acid, RNA from exosomes, RNA from circulating tumor cells, fresh or FFPE RNA. We've optimized some of the protocol to be especially compatible with RNA isolated from FFPE samples. It's one of the reasons our amplicons are so small, because it makes it more compatible with FFPE. So you're good and you're golden if you've got RNA from various sources, including FFPE samples. All of these are perfectly fine up front of QIAseq RNA. And so, again, from sample isolation through targeted enrichment and library construction, along with the adapters and such to run on the NGS platform, along with the primary and secondary data analysis and a link for Ingenuity pathway analysis, you have the complete system to do gene expression analysis and gene counting with this product. Here's the workflow in a very simple way. We're doing first-strand cDNA synthesis. We do a first primer extension on that cDNA synthesis where we add the molecular tag. So the random molecular barcode is added after random prime cDNA synthesis. We do a very aggressive cleanup to make sure that that gene-specific primer extension molecular tag is removed. We do a limited number of PCRs with the gene-specific primer from the other side and the adapter sequence we added earlier. Then we do a universal PCR, put on sample indexes, clean it up, and we're done. We do need to quantify the libraries so we can mix them together and put them on the sequencing instrument. And this works in a 96-well plate format as well. Everything that you need except the reagents that are used to run the sequencing platform are included in the kit. So a little bit more detail. We make our cDNA. We have a panel of gene-specific primers that have the random molecular barcode and a universal sequence on it. We do a single primer extension, clean it up, come back and do a very limited number of PCRs with a gene-specific primer from the other side, which has an adapter on it, along with a universal primer RS2 from the first side. So if you notice, the gene-specific primers are never in the tube together at the same time. And then we're doing universal PCR that's adding the sequencing adapters and sample indexes. So if you think about what you get at the end, if you've got 5 copies of a PCR product with or without sample indexes, if you don't have sample indexes and you read 5 copies, you know you have 5 copies of a transcript, and that would be what you'd see here on the left, 5 replicates. But if you do have the random molecular barcodes in there, what you'd have is what you have here on the right. You would know that you have 5 unique RNAs that were copied into cDNA and then read on the machine. And these 2 scenarios are very different from each other. In one case, you've read the same transcript or different ones. You don't know 5 times. But in this case, you know that you captured 5 different molecules. And statistically, those 2 things are very different. This is just an example of the robustness of looking at different plex libraries with different input amounts of RNA. This ranges from 12 plex to 1,000 plex, so 12 genes to 1,000 genes. From a nanogram of input RNA to 20 nanograms, and you can see the specificity of the libraries are all 97, 98%. And each of these libraries was sequenced to a depth of 1 million reads. So it doesn't matter whether it's a relatively small gene panel or a large gene panel, whether you're using a nanogram of RNA or 20 nanograms of RNA. The nanogram of RNA is actually, and the small panel is actually more challenging. You need to do a couple of more cycles of PCR to make enough material. You would notice the difference in these in terms of the barcodes. So a 12-plex library with a nanogram would have a lot less unique barcodes than a 1,000-plex library with 20 nanograms because you're sampling less RNAs. This is just an example of reading raw counts versus reading barcodes on ERCC standards that are spiked into universal reference RNA. This was published by a colleague of mine who actually sits in the office next to me, Bill Wang, in BMC Genomics last year. The theoretical CV based on amplicon counts, normalizing against barcodes versus just raw reads, and you can see that you do correct for and get low CVs when you use molecular barcodes. And if anybody is interested in this paper, we can provide that reference for you. So let's just look a little at performance. This is an example of a 288 gene panel that's run on 10 nanograms of total RNA. And it's run on 2 samples, human brain and human universal reference, which is a pooled RNA sample. And it's run on the ION torrent PGM, and it's run on the Illumina MiSeq. And we're looking at the fold difference between these 2 RNAs on the ION and the fold difference on the MiSeq and comparing those numbers to each other. And what you can see is that the data is platform independent. So the gene expression levels that we see in these 2 RNA samples, even on these 2 completely different platforms, one is emulsion PCR and electrochemical sequencing, and one is sequencing by synthesis, and all this stuff is essentially the same, with a 0.97 correlation between the 2, which is really pretty remarkable. This is interlab precision between our lab in the US and our sister lab in Hilden. The same panel, the same samples, but 2 different people, 2 different instruments, 5,000 miles away, Again, the correlation is 0.98, so both of them run on two different MySeqs in two different places. And again, fold changes are essentially the same. This is interesting. This is when we first got our NextSeq. This is with a beta customer who was using, we were both using our NextSeqs for the first time. It's a 384 gene, custom gene panel that we made for this beta tester. And this is their data versus our data. And you can see, in fact, it's exactly the same. And we were both totally new at using this instrument. Again, on the reference in the brain RNA, and our correlation between the two labs is 0.999, which is just totally remarkable. So technical replicates, I don't think you really necessarily need them. Biological replicates make more sense. This is interesting. This is old data from whole transcriptome from Illumina compared to this 384 gene panel. So they're both done on Illumina machines, different people, different years. And in fact, our differential expression using this panel versus Illumina data done on, I don't even know what instrument that was done on, but it was done by Illumina and published on these two reference RNA samples is essentially the same. So the panel differential expression versus whole transcriptome expression and exhaustive bioinformatics essentially comes out the same. And then one last comparison, and this is looking at gene expression digital NGS versus qPCR. So again, the same universal human versus the universal human brain reference, and it's 384 genes, and we did this by the sequencing on the MySeq, and we did it by PCR and singleplex PCR with cybergreen assays that we had in-house. And the PCR was normalized by delta CTs, and we used housekeeping gene controls. The RNAseq is normalized by unique barcodes versus total number of unique barcodes per sample. And you'll hear more about that in an upcoming seminar, how we do the normalization. And so if you think about it, the NGS required 5 nanograms of total RNA, and the qPCR experiment required 1,200 nanograms. So the qPCR was 3 384 well plates and 1.2 micrograms of RNA, and the NGS was 5 nanograms. So a lot less sample. And in fact, this is just log 2, so it looks a little bit messier, but it's only because it's log 2. Same data. This stuff is for real, and it does work. And it's fairly robust, instrument to instrument, user to user, platform to platform. Just some sensitivity benchmarking with ERCC. So ERCC are industry standard calibrated RNA standards. It's a bunch of different transcriptionally synthesized controls that are spiked in at known copy number. These are just added to a 384-plex panel and added at from 86 to 700,000 copies. And there's multiple ones at different copy number. And there were 3 technical replicates. Yeah, I know sometimes you run technical replicates. And so what's interesting here, if we look at the left side, you can see molecular tags captured per target. So that's how many unique targets we captured versus the input amount. And you can see things fall apart a little bit right about where we hit about 100 copies. And now we could have sequenced this deeper and probably firmed up those numbers, but it's an interesting point because if you look on the right side, it pretty much says the same thing. If we look at the CV versus the molecular tags that are captured, things fall apart about the same place. When we hit about 10 unique tags per transcript, the CVs go up. So this is where that number comes from that I mentioned before, that when we capture less than 10 unique transcripts for a gene in the sequencing experiment, our CVs tend to degrade. All of this translates to about a threshold of accurate quantification in this experiment down to around 100 copies of an RNA in 20 nanograms of total RNA. And that's the equivalent of about 0.2 copies per cell, or 1 copy for every 5 cells. You can boost that sensitivity by reading, using more read budget and reading deeper. If, in fact, that your average reads per molecular tag were very low. And I'm going to show you that on an upcoming slide. So if you've read it exhaustively, if you've read every molecular tag there is to read, you'll know that from the data output, and I'll show you that. So this is just an example of the effects of sequencing depth on sensitivity. So how do I know that I'm reading it deep enough? And so that these are just, actually, I think these are tenfold difference in depth. And what you see is essentially things are pretty much unchanged, except at the low end. And what you see is the signals bottom out for the samples that are read at lower depth. So things flatline. And then it's a repeated thing that you see over and over again, that the lowest, things that are expressed at the lowest level tend to flatline if you read it at a lower depth. And that's expected. But what this means also is that if you want to economize and you're reading a 384 panel, and you have a choice between possibly losing sensitivity at the low end and reading 5 or 10 times as many samples per run, you might make that choice and say, rather than reading only 10 samples, I'm going to read 100 samples per run and sacrifice the bottom 10 genes. That's a choice that you can make. So let's just turn to data analysis. When you get a FASTQ file off your instrument, you come to QIAGEN's website and you upload that FASTQ file. Once you've uploaded them, you do an RNA quantification job. You create a job. You pick the file that you're using. You come back after 2 cups of coffee. What the software is doing it's identifying in your sample the molecular barcodes. It's stripping them off and holding on to them. It's stripping off the primers. It doesn't use the primers to align your sequence to the genome. It uses the sequence next to the primers. So it actually, when it maps your read to the genome, it uses the actual internal sequence. It wouldn't be fair if it was using the primers to align you to the genome. So you'll see things in your report that say off target. What it means is that a primer may sit down in the wrong place in the genome and the sequence next to your primer is not right. So once it confirms that it's the right sequence and it's the right size, it's then going to go and essentially mate the molecular barcodes back to the sequence and count how many unique barcodes are associated with that sequence. So you get a summary of your data. These are 3 different samples. And you'll see total reads per sample. So of your, this is on a MySeq, so of the 12 million reads that were on the MySeq, I just grabbed a couple of samples here. So these are 3 samples of 3 million, 2 million, and 2 million reads. There are some reads that are dropped here. These are smaller than 55 bases, so these are probably just some primer-dimer contaminations, a very small number of reads. And there are various reasons some other reads get dropped. So you can see there's a couple of off-target reads here that get dropped. Some get dropped for other reasons. It's not terribly important. You can see that the number of reads that pass all filters, for example, in this sample was over 3 million. And what this says is, of these 3 million reads, there were 952,692 unique molecular tags. So you read 3 million reads, and on average, each unique RNA was read 3 times. In this sample, each unique captured RNA was read 2.4 times, and this one 2.5 times. And that's a reasonably good number. You don't gain very much by reading deeper. If you read those samples twice as deep, that number may not change very much. It may go up, that number would change. If your reads per molecular tag was 1, chances are if you read it twice as deep, the number would not be 2. It wouldn't double. So it would mean that there were molecular tags you were missing. This number goes up until around 10, and then it plateaus. So yeah, you're missing some molecular tags here. It's only going to have an effect on the lowest abundance targets. And the higher this number is, the more diminished return you get by reading deeper. So we generally aim for this number to be around 2 to 3. And that's sort of a happy medium for using our read budgets correctly. So you can see here that we're using about 97% of our reads, which is pretty typical for counting. So this is pretty much what we expect. If we look at the details, this is a pretty big gene panel, so I only show you a part of it here. Gene IDs, gene symbols, which strand of the genome it's on, chromosome location, what genome build. A lot of our amplicons are in single exons. It's easier for us to do to look at PCR efficiency for an amplicon that way. Because we do that, we include genomic DNA controls that are spread out on the genome. As in what you can see here is that the genomic DNA have a couple of counts. These are transcriptionally dead. They typically have 0 to 20 counts, and you can see here on these 3 different cDNAs, they have 0 to 20 counts. The software will flag any gene in which your counts are close to the genomic baseline, which is the mean of these numbers per sample, as being sort of unreliable. So if the mean here is 15 and you have a gene whose tag count is 18, it's going to flag it, and that's very, very close to background. But you can see for all these genes here, they're all way above that number. So this is all real. These are unique tags gene by gene. So for example, XRCC5 had 8,246 tags in this sample, unique transcripts that were captured. We do include a bunch of reference genes, which we have in there because over all of our experiments, and from databases, they're expressed at reasonably moderate levels and fairly wide range of expression. You're free to use those as reference genes. You're free to pick among them. You're free to pick any genes in your panel that you want, or none of them. If it's a diverse panel, we typically use total number of tags as a normalizer. If it's a targeted panel where all the genes are differentially expressed, you probably don't want to do that. You probably want to use a reference gene, and you can add whatever you want to a panel. So unique barcodes divided by total barcodes, housekeeping genes, one of them, some of them, all of them, genes of your choice, whatever you like. And this is all done on the secondary analysis. You label your samples as control, as group 1, group 2, whether they're replicates or not. And then you're going into the data plotting region where you're doing heat maps and scatter plots and volcano plots. You don't have to do this here. You can just take your primary data analysis and leave with the unique tags per gene and do your own data analysis. You can take the raw data and leave, or you can generate the kind of plots that you're used to generating for differential gene expression analysis. And if you've got replicates, then you can get p-values and such. So I just want to wrap up a little bit here and say what we're getting out of this system, this turnkey system, is sensitive and targeted expression profiling for you at less than 1 copy per cell. It's extremely flexible in design that lets you look at 12 targets to 1,000 targets or more from 1 sample, and that should actually read to 384 samples. Specificity is maintained, so you're not wasting read budget on off-target things. The uniformity of the assays, which is, it's difficult to see in RNA, but we know that it's extremely high. So the assays work extremely well together. The molecular barcodes remove all PCR bias. So you're not counting anything to do with PCR bias. You're counting unique captured molecules. You don't have to do anything weird to your RNA. You're just using total RNA, 1 nanogram to 20 nanogram. You can tailor your experiment very closely to the read budget on your instrument. It works very well with FFPE. Some FFPE samples are terrible, and the molecular barcode will show you that. And you'll know which ones to look at and realize that they're so bad you probably shouldn't be using them. We have a lot of know-how on content, and we can help you with that. And the workflow is as easy as it possibly can be for either a novice user or for an experienced user to go from that RNA sample to those fold change and differential gene expression analysis. So webinar 2 is going to be a deeper dive into the workflow and the data analysis. So I just brushed upon this. Raed will talk a lot more about how to actually set that experiment up. There are some special handling considerations for FFPE, and that actually should be RNA, cell-free RNA. There are some considerations about QC of samples, QC of libraries. There are some platform-specific considerations. This is just a chart that I'm sure that he'll show you again. Some rough calculations on how many genes and how many assays can run on an instrument. So, for example, we typically run 96 genes. We do 96 by 96 on a MiSeq. We do 384 by 190, or 96 on a NextSeq. What our multiplexing capabilities are on the instruments that we have. And then webinar 3 is interesting. It's a joint webinar between a scientist in my group who's been doing applications work and doing cell culture and other expression analysis experiments, along with an expert from Ingenuity on using pathway analysis. So when you use one of these panels, you get to upload your data to Ingenuity and do pathway analysis, and it'll be fun to listen to somebody who's an expert at this talk about how to make that bridge between differential gene expression and using the Ingenuity database to do that analysis. Finally, just to show you what these products look like, the panels are either 12 or 96 samples for our essentially library panels, which are pre-developed content. You're allowed to modify these panels by adding up to 25 additional genes. So if we have a panel you like, it's cost effective to add some genes to it. You can do that. If there's something entirely out of the blue that you want to make, you can do that too. And then we just have the indexes for the ION system and the Illumina system. And there's a library quant kit, so if you want to skip the bioanalyzer to normalize your libraries, you can use a PCR-based system. There are some panels that we have specifically designed for sequencing. These are from 250 to 500 genes at a time. But all of the content that we've developed over the years for PCR-based analysis are also available to make for NGS. So there's over 200 panels that are 90 genes or so that can be translated into NGS. So the guys just wanted to show you where we are. QIAGEN headquarters is in Germany. QIAGEN U.S. headquarters is in Germantown, Maryland. Of course, a German company would have its U.S. headquarters in Germantown. My NGS group is up here in Frederick, which is about 15 miles from Germantown. It's the front of our building here. And in this group, there's a technology development group who do early feasibility on cutting-edge technologies that are run. It's run by Bill Wang, and Quan Pang and his group developed the initial technology feasibility on the barcodes on RNAseq. All of our bioinformatics is run by John DiCarlo, and these two gentlemen and his group developed the RNA analysis tools for us. And Chung, Matt, Melanie, and Jeff are the people in my group that did all the product development for this work. And with that, I think I'll turn it over to Christian and let him field some of the questions. Christian?
Thank you very much for that.
Yeah.
Thank you very much for the very informative presentation, Eric. That was really, really great. Before we start on the question and answer session, I would like to remind the audience to how to submit your questions. You can submit your questions by typing them in the questions box that appears on the lower right-hand side of your monitor. And with that, I'll start off with the first question. The question is, if there is no polyase selection, can this technique be used on samples with genomic DNA contamination?
Yeah, so there is a recommended DNA step on the RNA, and that's why we're including a panel of transcriptionally dead DNA controls. On all of the DNA, all the RNA samples we've looked at, we typically get anywhere from 0 to 20 counts for that panel of genomic DNA. The analysis software will take a mean of that number, and if any of the counts on an RNA target are within twofold of that number, and remember this is quantitative, so if it's within twofold, it'll flag it and let you look at it, and so you have a choice. So if the DNA number is say 15 and your target is at 30, you probably have a low level of expression for that target in your sample, but it's probably not statistically an accurate number. Normally, if you have 10 unique tags and the genomic DNA number is 0, I would believe those 10 tags. But if you're within 10 tags of the background and the background is higher, I'd question it a little bit, and you can go back and be a little more aggressive with your DNase treatment. In general, it's not an issue. Most of the time, the DNA number is 0, so it's generally a non-issue.
Okay, thank you, Eric. The next one is: would this system work on ION torrent system?
Yeah, we routinely use it on the ION torrent. The sequencing on the ION torrent is quite good, and the single-ended reads on the ION are long. It's fine. Yeah, the read budget's a little smaller, but it's fine.
The next question is regarding the sensitivity of this system. Will this technology be able to detect copies less than 10?
Probably. We can easily detect between 10 and 100 it depends. So the variables are how much of the read budget you give per sample, and the mix in the panel. So if you have a panel where all of your targets are relatively rare, the sensitivity will be higher. The funny thing is, compared to PCR, in PCR you've got to split your sample to all of your assays, and in NGS you don't, right? But in NGS, one assay sucks up part of the read budget so that it's not available for your other assays. So if I had a rare target in with a bunch of abundant targets, the abundant targets would suck up more of the read budget. So if you're careful and you design a panel so that you don't mix targets that have extraordinarily high dynamic range with each other. For example, you don't put GAPDH and actin and cyclophilin in the panel with your rare targets. You will increase the sensitivity of your panel. And so, yeah, you can detect 10 targets. We just haven't bothered to do that. So we have panels that have one target will have 75,000 or 100,000 tags, and another will have 20. If you really wanted to detect things that low, you ought not put that really abundant thing in there with it. And there are databases that describe relative abundance of every gene in the transcriptome in different cell types that you can look up and maybe decide that you don't want to put, globin in with your panel, for example. So yeah, 10 copies is not that hard. The capture efficiency if you have 10 copies of an RNA in a sample. So the other variable is how big is the RNA sample those 10 copies are in. Since the cDNA synthesis is not targeted, if those 10 copies are in a microgram, that's different than those 10 copies being in a nanogram, right? So you can expect to capture in 20 nanograms or in 10 nanograms, say 20% of the molecules. So if the 10 copies are in 10 nanograms, you have a much better chance of capturing them than if they're in a microgram. So there's a number of variables there. If you really need to capture them from a much bigger sample, like when someone's trying to pick up a rare virus in a blood sample, that's when people do targeted reverse transcription with a gene-specific primer. Yeah.
Okay,
Sorry.
thank you.
Yeah.
That's good. That's great.
Yeah.
The next question is, do we still need to validate the data by qPCR?
The only time I have seen a disagreement in the data between what we're doing, I would say the answer to that is no. Although an editor of a journal might disagree with me. The only time I've seen two instances where our data has disagreed with whole transcriptome data, and one of them was where we had designed an assay against a gene with a very complex splice pattern where we knew we were missing a splice form. And we underestimated the level of expression of a gene in a particular tissue. And the other example, which was recent, was a collaborator found an odd example with a histone gene, and he said that they do behave strangely in that particular gene in targeted sequencing. But I think, you know, when people first came out with hybridization arrays, editors always made them confirm it with PCR, and then they stopped. And when people first came out with PCR, editors made them confirm it with northern blotting, and they stopped that too. So I think it's just a matter of time that people believe this. I think it's extraordinarily accurate.
Okay.
Next. Yeah.
Okay. Great. The next question is this technology compatible with pre-amplification technology for small samples such as Repli-G?
Yeah, so we've done that, and what we find is that, I mean, we've done that from like single cells, and we find that the precision of it is not quite as good as not doing it. So if we took a nanogram of RNA and used Repli-G and then took a nanogram of the amplified material and compared it to a nanogram of unamplified, the trend is there, but the precision is not. I hate to say that, but I mean, if you have no choice but to amplify, then you have no choice. But it's not perfect. I think that the solution to that is going to be doing molecular barcoding on the single cell level, so that you're tagging every molecule at the single cell. And we're actually working on that, but we don't have that running yet.
Okay. Do you have any comments on the quality of the RNA samples you need for this technology?
The best observation I can give you is that the molecular barcoding is like an all-seeing eye. So, for example, you know, I know when people are looking at FFPE samples, there's ways to increase your yield of the library so that you can sequence it. So it's very routine for people to say, you know, I added 2 cycles, I used more material, I used 100 nanograms instead of 20 nanograms, I made, you know, and that kind of thing. But if you look at if you look at time after time at the samples that we're getting, we can easily make a library from FFPE. And when we multiplex those libraries on the instrument, because we're normalizing the library onto the flow cell, we're getting the same number of reads. So every library is giving us 2 million reads. But if you look at the barcodes, a fresh sample gives us, from a 2 million read library, will give us 1 million barcodes. And an FFPE sample from a 2 million read library will give us 5% of the barcodes. So what it means is, instead of sequencing each unique read twice and having a library of high diversity, we're sequencing each molecule 20 to 50 times, and we're just sampling the same small number of available molecules over and over and over and over again. What does that mean? I'm not entirely sure what that means. If it's not biased, it's probably okay. But when somebody takes a really small sample and says, I just amplified the crap out of it, and read it and all is good, you know, if they're, for example, if they're looking for a somatic mutation, and they said, well, I read 1,000 molecules. What may be actually true is that they read 2 molecules each 500 times. So the barcode lets you see things that you might not necessarily want to know about. So you certainly do see very clearly in particular from FFPE samples, that the amount of available nucleic acid for library construction is much lower than your bioanalyzer or your nanodrop says is there. It's not that bad from circulating nucleic acid, but the fixation process in FFPE is really a bad thing. Circulating nucleic acid, even though it's fragmented, is still available for library construction, much better than FFPE.
Okay. We are run out of time, but what I'm going to do is there are a couple of questions which are a little bit have some common themes around it. So the one thing is, what type of controls are included in the panel, and do you have any comments on the controls?
Yeah, so if you have a panel of genes which are involved in processes, genes involved in cancer, kinases, things like that, in general, and I've spoken to people who are other people who are experts at this, it's actually it works out really well to normalize the unique tags against the total unique tags, because some genes are unchanged, some genes go up, some genes go down. That average works out really well. But if you have a panel that are picked because they're differentially expressed in your system, you make a panel of all the genes that are overexpressed when you treat the cells with some reagent, you're going to have this large-scale gene expression change. The two samples are going to be so different from each other. You're going to need something invariant that you know is invariant. So then you really are going to want to specifically add something to your panel, some gene that you know can be a housekeeping gene in your experiment. And that burden is on you. We've added a number of genes on the panel that we have found to be quite invariant in a large number of tissue types in a large number of situations. It may not work for you. They're there. You can look at them and decide whether you want to use them. You can ignore them and pick something else from the panel. You can add your own gene to the panel. You can use a software such as Norm Finder. Norm Finder will suggest something from the panel. It's a question that people have been asking for 30 years. We picked some genes that won't use up much of your read budget, and we put them on the panel as a service. If you're designing your own panel, you're welcome to add them or add anything you like. So it's hard to say, right? And not everything will suit everyone. But for the panels that we sell, most likely the normalizing against tags is the reasonable way to do it.
Okay, the last question for today is, do you have any specific recommendations as to what instrument we should run it on, an ION torrent or an Illumina or a QIAseq?
I think that it depends what you have. We have certainly been doing this. The sweet spot for our lab has been the MiSeq. For us, because of the number of people in the lab and the size of the experiments they're running, that's been the sweet spot. We tend to have to save up experiments to run it on the NextSeq to actually coordinate enough experiments to fill it up. But because if you actually can fill the NextSeq, it's cheaper to run it because it has such a huge budget. But then the MiSeq's been very convenient. So we have 15 people in the lab that are running sequencing, and we have 2 MiSeqs and an NextSeq, and 2 QIAgen instruments, and 2 PGMs. And the MiSeqs run 24 hours a day. Yeah.
Okay, thank you very much. There are many more questions that have not been answered here today. What we'll do is we'll try to answer it via email. And also there are some questions regarding customization of panels and some pretty details. And what I would suggest is that attend the next webinar. We have the
that would be a lot of help. That would be a lot of help for them. Yeah.
Yeah. So that's what my suggestion to them, to attend the next webinar, which will be same time and next week. With that, I want to thank Eric once again for the wonderful webinar. And the webcast will be available for you for on-demand viewing very soon, within 48 hours. So all the attendees will receive the link to this on-demand video. So with that, I want to thank each one of you, and see you next time in the next week.
Thanks, everyone.
Bye.